When does a person become an addict? Is it in the minutes following that first hit off of a cigarette? Or does the first puff of marijuana open the gateway to a life of addiction? Could it be when a child sees their father crack open a beer as soon as he arrives home after work each day? Increasingly, the scientific community has become interested in the possibility that the seeds for addiction are actually planted in the first minutes of a person's existence, months before a person is even born. In this episode of Health at House of Commons, we'll be exploring how addiction can be influenced by genetic factors that begin to create the people we become from the very moments of our conceptions. One of the oldest arguments in the study of human behavior is the nature versus nurture debate. At its core, this dispute is based on the question of where we acquire the traits that make us different. Those on the side of nature say that a person's genetics are the root cause of how a person will act throughout their life. Genetic traits are acquired from our parents and established before we are even born. As you will see, it is quite clear that much of our identity, like our hair and eye color, are determined by our genetic inheritance. Traditionally, it has been much more difficult to evaluate the influence of genetics on things that we cannot see, like preferences for some foods, or the likelihood of developing some types of diseases. On the other side of the debate, those advocating nurture's influence contend that a person's behavior is influenced by their physical and social environments. The physical environment can include anything that we come into contact with throughout our lives. Some of these factors may be fairly easy to identify and could include the foods that we eat, the homes and cities that we live in, or chemicals that we are exposed to in the workplace. In many cases, our physical environment is less obvious and can include things that we can't see, like lead in the air and water. All of these things are capable of making changes to our bodies and in the way that we are compelled to act. The influence of our social environments can be even more complex. Our social environment consists of all the people that we come into contact with throughout our lives, as well as the ways that we interact with them. This could include family, friends, co-workers, and even strangers. It also includes the ways that we talk to each other and the ways that we are treated by the people that we meet throughout our lives. It may be fairly obvious that who we are is not determined by either nature or nurture alone. It would appear that the seeds of our identities are planted by our individual genetic makeup that is established at the time of our conception. This identity is modified over time by the people, places, and things with which we come into contact. Although the influence of the social and physical environments is undeniable, this lecture will focus primarily on the impact of genetics on our behaviors, and specifically on an individual's predisposition to becoming addicted to a drug or behavior. We will first have to begin by developing a basic understanding of what genetics are and how they work. We will examine where they come from and what makes your genetics unique. We will then explore the process by which your genes become the physical and behavioral features that define each of you. Once we have gone over these essential concepts within the study of genetics, we can then examine some of the research that has been conducted on the involvement of genetics in addiction. This will include not only what genes may make us more likely to become addicted to specific drugs, but also which genes may allow us to be more likely to respond well or poorly to different types of treatments. Finally, we'll finish up this class on genetics by taking a look toward the future. When studying genetics, one of the essential disclaimers is that almost everything that we know about genetics up to this point may become outdated within a matter of years. Nevertheless, a few areas of genetic research have received strong interest that we can say with some confidence that these areas will dominate many of the conversations about genetics for the next several decades. The first, epigenetics, concerns modifications that occur to the genome after we are born. We'll expand upon the subject shortly, but for now let it suffice to say that the field of epigenetics describes the effects of nurture upon nature, that is, the ways that the environment impacts your genome. The final topic that we'll discuss today is an emerging technology called CRISPR, a genome editing tool that may become the most transformative instrument that the biological sciences has ever seen. Stepping back, what is a gene? We've already used several terms that are similar, but they have slightly different meanings. The noun gene, or the adjective genetic, refer to a small portion of your DNA that encodes a specific heritable trait, like eye color, hair color, etc. The noun genome, or the adjective genomic, refer to the entire collection of genes that an organism, like a human being, possesses. Genotype is a term that describes the specific portion of a person's genes that are associated with a specific trait, whereas phenotype describes the observable trait. Again, hair color being an example of a phenotype. The different variations of a given gene are called alleles. For the gene that determines eye color, one person may have an allele that gives them blue eyes, while another person possesses the allele that gives them brown eyes. DNA, deoxyribonucleic acid, is the language of the genetic code. DNA consists of 4 different units: adenine, guanine, cytosine, and thymine, that are called nucleotides. Nucleotides are a combination of a sugar, called deoxyribose, and a nitrogen-containing compound. Each of these nucleotides is bound together by a phosphate molecule in a chain-like structure of 2 intertwined spirals, commonly referred to as the double helix. A string of 3 nucleotides in a row is called a codon. As we'll see shortly, each codon is associated with a specific amino acid. Amino acids can be stringed together to form proteins, which are the way that your genes are expressed as physical traits, whether it be something easily visible like eye color, or something that we aren't able to see with the naked eye, like whether or not someone is diabetic. From this point onward, I should note that I will specifically be describing human genetics. Although the process of genetic duplication is fairly similar for non-human animals, as well as plants, some specific details may be slightly different. With the exception of the gametes, the sperm and the egg, also called ovum, each cell in the human body contains DNA in the form of 46 chromosomes. All cells other than gametes are referred to as autosomal cells. Of these 46 chromosomes, 23, half of them, come from the father, and 23 from the mother. This occurs through the processes of meiosis, fertilization, and mitosis. Without going into too much detail, meiosis is the process in which a human cell with 46 chromosomes first divides into 23 chromosomes, and then divides again into sister chromatids. The gametes will contain these sister chromatids, essentially 1/4 of the genetic information contained in each of the original cells. It is also important to understand that during each of the 2 phases of meiosis, some genetic rearrangement called crossing over occurs. During this recombination period, the chromosomes are reorganized to produce a genetically unique gamete. To put this into somewhat simpler terms, each sperm and egg contains genetic information from each of a person's parents. The result of this is that a child is born with the genes of not only each parent, but each grandparent. So, a person's single sperm or egg is not a clone of the genes that this person received from their own parents. To put this process into some perspective, The number of possible ways that a person's chromosomes can be arranged during meiosis allows for approximately 8 million genetically unique possible gametes. This means that a man has the statistical opportunity to create 8 million unique sperm and a woman to create 8 million unique eggs. When any given man and woman's gametes combine together during fertilization, there are almost 70 trillion unique combinations that occur from these two people alone. During fertilization, the gametes combine together and create an autosomal cell with 46 chromosomes. This cell will separate through a process called mitosis and will eventually develop into a complete human being. Moving onward, let's examine how a gene is expressed as a physical feature. The first phase of this process is called transcription, during which DNA is processed into RNA, ribonucleic acid. In the second phase, called translation, RNA is converted into proteins. These proteins will then serve as the tools that directly or indirectly contribute to all of a person's characteristics, whether it be eye color, height, whether a person is lactose intolerant, etc. During transcription, the double helix of DNA is split apart and the sequence of nucleotides is copied onto a complementary strand of RNA by an enzyme called RNA polymerase. An enzyme is a specialized protein that performs a specific function. RNA is almost identical to DNA, except that whereas DNA is double-stranded, RNA consists of only a single strand of nucleotides. In addition, in RNA, the nucleotide uracil replaces thymine. The primary tool of the process of translation, in which RNA is converted into proteins, is called the ribosome. A ribosome is composed of some RNA and several proteins. Its function is to recognize the 3 nucleotide codons on a strand of RNA and to bind together their corresponding amino acids in the same sequence that the codons occur on the RNA. As hinted at earlier, a string of amino acids is called a protein, and unique sequences of amino acids result in the creation of unique proteins with many different functions. All of this is fascinating stuff, and we could probably now spend the rest of the day, or probably even more realistically, the next year, discussing the specific mechanisms involved in the processes that I've just summarized in a little more than 10 minutes. The question that we must now move on to, however, is how do these concepts relate to the development of addiction? Is there something happening at the genetic level that makes one person more likely to want to do a drug over and over again, whereas another person will be able to exercise more control over whether or not they want to use a substance more than once? Another question we'll address is whether genetics may be able to predict which people will respond better to pharmaceutical treatments for addiction. Much of the knowledge that we have about which genes are associated with specific traits come as a result of the Human Genome Project. The Human Genome Project was the largest biological study that has ever been conducted. Taking over 13 years, this international study managed to identify each of the 20,500 genes in the human genome. Much of the genetic research conducted since then has tried to determine the functions of each of these genes and the variations of each of these genes. By examining the traits of individuals, it attempts to identify linkages between variations of each gene with each specific trait. There is an essential disclaimer to making allegations about the genetic causes of behavioral disorders. While a single gene may be implicated in some health problems, for example, cystic fibrosis, Huntington's disease, and sickle cell disease, single gene diseases are fairly rare. Furthermore, it is extremely difficult to identify individual genes as the source for psychiatric or behavioral disorders. The general scientific consensus is that Similarly to extremely complex diseases like cancer, these are often the results of complex combinations of genetic and environmental factors. In essence, this means that no one gene is going to make a person an addict, but that combined with other genes, and with the contribution of a person's living conditions over the course of their life, a single gene may make a person more likely to become addicted to a substance. This is all beginning to sound like a long-winded way of saying that we aren't able to identify genes that make a person more susceptible to developing a substance use disorder, but that isn't necessarily the case. It's just important to understand that this is a field of science that is still being developed, and the enormous number of possible ways that a person's genetics and their environment can be combined make it very difficult to make a definitive assertion about many of the current findings that science has produced related to genes and addiction. So what can we say? Let's begin by looking at the research that has been conducted on which genetic factors make a person more likely to develop alcoholism. Although some preliminary research has been conducted on other drugs, the availability of alcohol in our society and the prevalence of alcoholism throughout our population have made this substance the most commonly studied drug by far among genetic researchers. I suspect, and I think that you may find that you agree with me, that many of the findings that I will discuss could probably apply in some ways to other drugs. Most researchers just haven't studied other drugs as extensively because other drug users may make up a smaller portion of the population and therefore may be harder to find. In addition, the users of illegal drugs may be less likely to be willing to come forward out of fears of legal repercussions for admitting to illicit drug use. Lastly, legal research into illegal drugs is highly controlled, and it is considerably more difficult to obtain substances like cocaine, opiates, etc. Within a scientific environment. Some of these genetic studies into alcoholism have focused on the genes that control how alcohol is metabolized differently by different individuals. Metabolism, in a nutshell, is how one chemical compound is broken down into other chemical compounds. The metabolism of alcohol occurs through 3 stages. The first stage begins with ethanol, which is the proper term for all the types of alcohol that we drink, whether it be wine, beer, or liquor. Alcohol is a much more general term in chemistry and also includes substances like methanol, also called wood alcohol, or isopropyl alcohol, which is a general term for rubbing alcohol, which are both considerably more toxic than the alcohol sold in liquor stores. So again, we begin with ethanol, and ethanol is metabolized by an enzyme called alcohol dehydrogenase, or ADH, into a compound called acetaldehyde. Acetaldehyde is fairly toxic, and it's the chemical that causes the unpleasant symptoms of alcohol use, such as facial flushing, sweating, headaches, and in severe cases, heart problems, unconsciousness, and convulsions. Fortunately, in most people, acetaldehyde is further metabolized by a second enzyme called aldehyde dehydrogenase, or ALDH, into a third compound called acetate. Acetate is very similar to acetic acid, which is the name used in chemistry for vinegar. Acetate's going to be the metabolic end of the line, and that's what will be excreted in the urine. So we have 2 genes that regulate this process that have been linked with the development of alcoholism. Both of them are associated with parts of the process that produce lower levels of acetaldehyde, the chemical that, again, causes most of the unpleasant effects of alcohol. The first gene causes our first enzyme, ADH, to function poorly. This means that less acetaldehyde is produced, or that it's produced at a slower rate, meaning that a person keeps a higher level of ethanol in their system for longer. The second gene causes the second enzyme, ALDH, to work even better than in most people. For people that possess this gene, the ethanol has already been converted to acetaldehyde, but our second enzyme converts the acetaldehyde very rapidly to acetate. In a nutshell, a person with either or both of these genes is less likely to experience as many of the negative effects of alcohol, and is less likely to be discouraged to consume large quantities of alcohol as frequently. The genetic links between alcohol metabolism and the development of alcoholism are fairly well established. Other connections between genetics and addiction are somewhat vaguer. Some studies have looked at the performance of nerve cell receptors that are activated by alcohol. And while we may have fairly strong evidence that people that possess specific variations for these receptors are more likely to develop alcoholism, we may not have a very clear understanding of how this works, unlike the cases of alcohol metabolism where we know exactly which enzymes are involved and have a pretty clear picture of how their functioning affects the development of alcohol addiction. For example, GABA receptors are activated by alcohol and are involved in processes that lead to sedation or feeling tired, a decrease in anxiety, impaired motor functioning, and symptoms of physical withdrawal. In animal and human studies, we've seen indications that people with one variation of GABA receptors are much less likely to be sedated by alcohol, and because they aren't going to feel as tired, they're much more likely to want to stay up and binge drink. Dopamine receptors are also activated by alcohol, and the result is the release of the neurotransmitter dopamine, which triggers rewarding effects. Serotonin receptors, on the other hand, are associated with compulsive craving and are more likely to make a person want to consume alcohol if they haven't been. Mu-opioid receptors are another neurotransmitter receptor that are activated by alcohol, and the result in this case is the release of endorphins, which are the body's natural painkillers. Endorphins, like opiate drugs, such as morphine or heroin, produce feelings of euphoria and analgesia, the reduction of the sensation of pain, as well as the negative effects of the sensation of physical withdrawal. Shifting gears briefly, we can see that genetic variations in neurotransmitter receptors appear to affect addiction to other substances other than alcohol as well. Endocannabinoid receptors, for example, are activated by marijuana. Some variations in these receptors are associated with higher rates of addiction and more severe symptoms of withdrawal and craving. Nicotinic acetylcholine receptors, on the other hand, are activated by nicotine and are involved in many processes throughout the body. People that are genetically coded to metabolize nicotine faster, unsurprisingly, appear to smoke more cigarettes per day. Studies in mice have also indicated that some genetic variations of nicotinic acetylcholine receptors appear to make the animals more sensitive to the rewarding sensations of nicotine, and thus make them more motivated to seek out the drug. Returning to alcohol addiction, but looking at the bigger picture, one study has found that a genetic variation caused a change in the paracingulate and caudate nucleus of the brain among people that consume more alcohol. This is an area that's involved in the perception of taste. And so this research indicates that people are more likely to become heavy drinkers if they actually taste alcohol differently than people without this genetic variation. The concept of a genetic link to substance abuse disorders is made all the more complicated by the question of what to do if a genetic cause for addiction is found. If you could test your children for these genetic differences and it turned out that they possess genes that made them more susceptible to becoming addicted to a substance, what could you really do? You might advise them to avoid that substance, but how is this any more effective than what you might do or say without any knowledge of your child's genetic makeup? Society's ethical dilemma is probably even more difficult. If a person is genetically predisposed to becoming addicted to a substance like alcohol, should there be a law that forbids them from obtaining it? This would almost certainly be considered an infringement on a person's freedom and would probably be grounds for a discrimination lawsuit. And again, keep in mind that there are not currently any single genes that are known to work by themselves that cause a person to become an addict. These genes work with other genes and must be combined with factors from a person's social and physical surroundings. So, a person can possess genes that make them more likely to become an addict, but without the right environment, they might never become an addict. You've probably all heard some variation of the saying, all it takes is the right girl and the right friends and the right bar. One area of genetic research seems like it will be more practical. Rather than trying to determine which genes make a person more likely to become an addict, these studies investigate which genetic variations may predict how a person responds to drugs used to prevent addiction, or, I guess in other cases, to cure addiction. This method of using genetic information to predict a person's likelihood of responding to a pharmaceutical treatment is called pharmacogenetics. Some studies have looked specifically at how well people with specific genetic variations of their mu-opioid receptors respond to naltrexone, a drug that reduces the response of opiate receptors in the brain. People suffering from alcohol use disorders with some genetic variations of their mu-opioid receptors appear to respond much better to drugs like naltrexone than other people. Other studies have looked at how people with genetic variations in their GABA receptors respond to topiramate, a drug that reduces sensations of craving. Similar pharmacogenetic studies have found genes that are associated with nicotine patches. This research has indicated that people that metabolize nicotine slowly are more likely to suffer from the effects of nicotine toxicity, which is going to give them unpleasant physical feelings when they're given a large dose of nicotine, as with a nicotine patch. And these people are less likely to continue with nicotine replacement therapy. Research like this may prove to be priceless in how addiction is treated in the future. Clinicians will be able to prescribe specific drugs with much more certainty for whether or not they will help a person overcome their addiction. Developing a deeper understanding of why some people do not respond to these drugs may also help drug developers create new drugs that address the needs of people that do not currently respond well to existing drugs. As of 2010, there were 6 companies offering genetic tests for factors related to addiction. 23andMe, Decode, GenePlanet, Biomarker Pharmaceuticals, MyGeneProfile, and Lumigenics. The most common test is for nicotine dependence, followed by tests for the alcohol flush response and alcohol dependence. The least common available tests investigate which variation of mu opioid receptors people have, which again predicts how likely a person is to respond to naltrexone or to become addicted to heroin or other opiate drugs. These tests should be considered with a fair amount of skepticism. As we've discussed, there's a lot of evidence that needs to be recovered for many of the links between genetics and addiction before it's really conclusively accepted by the scientific community. It might seem slightly frustrating that there really aren't many ways that genetics can be used to treat substance use disorders in the present day and age. That said, we may very well see some of these ideas start to emerge in clinical practices more and more over the course of the next decade or so. The technology needed to identify the genes associated with specific qualities of addiction already exists. We just have an enormous amount of information to sift through before we really clearly understand it. Recall from earlier that you are one of 70 trillion possible genetic variations of the child that your parents could have had. As you can imagine, it's expected to take quite a long time before we fully understand how each of the variations of each of the 20,500 genes in the human genome affects a person's individual existence. Adding to the complexity is the emerging concept of epigenetic modifications. Epigenetics describes the process by which a person's genome is modified over the course of their life. There are a number of ways that this can occur, but the most common, and frankly probably the simplest, appears to be DNA methylation. During methylation, a methyl group, which is a very small chemical structure, attaches to the genome. This methyl group can interfere with normal genetic functioning. The most common way that this occurs is through a process called gene silencing, in which methyl groups prevent DNA from being transcribed to RNA. This process is associated with many different types of health and behavioral changes, among them alterations of how the brain's reward system functions. And in short, epigenetic methylation may make it harder for the brain to produce dopamine, making it necessary to take even greater doses of a drug in order to achieve the desired reward response. This can lead to an accelerated rate at which a person becomes addicted to a substance. Unfortunately, epigenetic modifications are not limited to a single person. They can be passed on to one's children. Many epigenetic modifications appear to be the result of environmental factors that we are exposed to. Among these could be lifestyle habits like diet, exercise, or substance abuse. If there is an important lesson to take away from the concept of epigenetics, it may be that your decisions today to use a drug could affect your unborn children's entire lives. We'll wrap up this discussion of genetics by mentioning the hot topic in biology for the past couple of years, the CRISPR-Cas system. Typically just called CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats. Don't worry, I won't be repeating that again. This new technology was discovered in the RNA of bacteria in 1993. It was not until 2012, however, that it was discovered that it could be used to make very precise edits of the genetic code. If you've heard of genetically modified organisms, or GMOs, you may have realized that it was possible to edit a plant or animal's genes before now. The process of changing the genetic code has traditionally been a fairly imprecise process, though. Typically, genetic material was simply introduced from one type of animal or plant into another by inserting it into a cell containing DNA in the hopes that it would just sort of find its way to the desired location. Genes might be introduced by injecting them with a very small syringe, or they could be delivered inside of a virus, or they might even be shot into a cell by a tool kind of unsurprisingly called a gene gun. This general process, called genetic engineering, would usually produce many, many mutants, which are animals or plants with genetic variations that would not have been passed on through normal reproduction. And traditionally, only those mutants that achieved the desired results would be kept for future use. What makes CRISPR unique is that it is able to very selectively replace specific sequences of DNA. Rather than using the shotgun approach to genetic engineering, CRISPR makes it possible to identify a very specific genetic variation and replace it with the exact gene that one wants. As said earlier, this is a very new technology. If we were to compare it to cars, we're not even at the point where we could compare it to the Model T. We're closer to the point where we've figured out how to make like a wood-burning engine that can move a box on 4 wheels forward. The main problem that researchers are faced with right now is that while CRISPR is considerably more accurate than previous forms of genetic engineering, it still makes a lot of errors. In 2015, the first research into using CRISPR in human embryos was conducted in China, and it was pretty universally condemned by scientists around the globe. In early 2016, a British team was given permission to begin modifying the genes of human embryos, but this was under the condition that these embryos would never be allowed to survive until birth. A number of very valid ethical concerns exist for genetically engineering humans, and many of these still need to be discussed before any extensive research is conducted on humans. While we would obviously like to be able to prevent serious illnesses that are caused by specific genetic variations, most scientists are fairly hesitant to allow anybody to move forward with this kind of research. And in December of 2015, the International Summit on Human Gene Engineering took place and established some of the basic guidelines for current research. The primary ruling of this meeting is that CRISPR will be allowed to use for the gene editing of cells that are not gametes, again, sperm or eggs. And this way we can work on developing techniques for using CRISPR to correct genetic variations that play parts in serious and well-defined illnesses like cancer and Parkinson's disease. But CRISPR will not be allowed to edit the cells that are passed on to future generations. So probably can't expect to see the emergence of a superhuman race where the people are 10 feet tall and don't feel pain, at least in the near future. What we may see over the course of the next several decades is the slow emergence of methods in which CRISPR is used to replace genes that cause severe, debilitating diseases. Knowing what we've learned today about the genes involved in the development of addiction, one of the questions that will arise is whether or not people will be able to alter their genomes in order to reduce their body's tendencies to become addicted to a substance. If this does become possible, we will also have to ask if this is something that we would want to do. In summary, it is fairly clear that genes play a strong role in the development of substance use disorders. But although the exact ways that this occurs is becoming fairly clear for some processes, the research has still been very inconsistent for many. This emphasizes the interplay between a person's genetic heritage and the influence of their social and physical surroundings over the course of their lives. From an ethical and practical perspective, it may be exceptionally difficult to ever use our knowledge about the genetic origins of addiction to prevent addiction from occurring, but by increasing our knowledge of the genetic components of addiction, we should expect our ability to develop more personalized treatments for people that allow us to provide more effective therapies for people suffering from addiction. As I said earlier, it would take much longer than we have here today to discuss the finer details of how genetics work, and what I've presented to you today is only a tiny fraction of what you could expect to cover in an introductory genetics course. I hope that I have, however, provided you with a slightly greater understanding of how we inherit the characteristics that define us, and how these traits emerge from our individual genetic codes. And again, I hope that it's fairly clear that many of the findings that were discussed today can also apply to many other forms of addiction. Just as a person's ability to break down alcohol into other compounds appears to directly impact that person's chances of drinking to excess, and how the rate at which a person's body processes nicotine seems to predict the success that this person will have when pursuing nicotine replacement therapy, so also an individual's ability to metabolize other drugs may play roles in influencing how likely that person is to becoming addicted to a drug or to succeed in different forms of treatment. Likewise, many of the neurotransmitter receptors discussed today, like dopamine, opioid, and serotonin receptors, play roles in the mechanisms by which many drugs, like heroin, cocaine, methamphetamines, and ecstasy work. Just as genetic variations in these receptor types may make a person more or less likely to become addicted to alcohol, they are almost certainly also a factor in a person's probability of becoming addicted to other substances and behaviors. I also hope that I have whetted your appetites to learn more about the topic of genetics and addiction. If you would like to do so, I would definitely recommend the books that I have referenced for this presentation. For basic concepts regarding genetics, I referred back to Neil Campbell's iconic textbook on biology. This book is the one that's used in most high schools and colleges around the country, and is probably the most popular biology textbook still used today. For information specifically regarding the genetics of substance use disorders and addiction, I primarily looked at a book called Genetic Research on Addiction, which was edited by Audrey R. Chapman. Although I have focused primarily on the scientific findings of such research, this book also devotes a great deal of attention to the ethical concerns for researchers, politicians, and everyday people that are raised by genetic research into addiction. Video clips that were included in this presentation came primarily from documentaries that you should be able to find on YouTube. Clips describing genetics and addiction came from the Open University series called Investigating Addiction. The animation depicting the process of fertilization was produced by Nucleus Medical Media. Videos illustrating the decryption of the genetic code by the Human Genome Project were from the PBS show Nova in an episode called Cracking the Code. Video clips about epigenetic modifications were also produced for PBS's Nova and came from an episode on the subject of epigenetics. The video clips describing the CRISPR-Cas system were produced by Dr. Feng Zhang's research group at MIT's McGovern Institute for Brain Research. I want to thank you all for taking the time to watch this presentation of Health at House of Commons. Have a good day and a healthy future.